Welcome back to Translating Proteomics. Today, I am delighted to welcome Dr. Jennifer Geddes-McAlister, who's an associate professor in the Department of Molecular and Cellular Biology at the University of Guelph in Ontario, Canada. Professor Geddes-McAlister did her master's work with Brent Selinger at the University of Lethbridge, her PhD with Jim Kronstadt at the University of British Columbia, and her postdoc with Matthias Mann at the Max Planck Institute of Biochemistry. She is a leader in applying proteomics and systems biology techniques to understand host pathogen interactions and is a strong voice for moms and proteomics. Some of Professor Geddes-McAlister's recent work focuses on how we can expand crop productivity and human health by better understanding the dynamics of fungal disease and resistance. In a similar vein, her lab is leveraging proteomics and systems biology to learn how we can engineer plants to efficiently produce small molecules used to treat human disease. This is a fascinating field of molecular farming. That's farming with a PH. And I'm speaking with Jennifer just before we get to see each other in person at the Human Proteome Organization's 2025 conference in Toronto. Um, I'm I'm sure we're both very excited to connect with researchers of all types and backgrounds and learn how the field's advanced in the past year. Jennifer, thank you so much for joining us and welcome to Translating Proteomics.
Yeah, thanks for having me. I'm super excited.
There's so many fun things to chat about. I'd like to start off by going back in time and understanding a little bit about how you got interested in host pathogen interactions in the first place.
For sure. During my undergrad, I was doing biochemistry and with a heavy lean towards biology, and started working at the Agriculture and Agri-Food Canada Research Center, which was really very much focused on crop improvement, food quality, food safety, and I was working on fungal pathogens, which I always found very fascinating because a lot of work is on bacterial pathogens. At that time, it wasn't that long ago, but long enough, a lot of kits and that were all designed for bacterial work and not fungal work. So I just really loved the challenge of it. And that's when I first started doing proteomics and really starting to look at things differently. So what makes pathogen a pathogen? Why is it infecting a cereal crop? How is the crop defending itself? All those dynamics, I found just very fascinating.
That's really interesting. So you're right. I mean, my own background, I started studying first a very esoteric bacteria called Pyrobaculum aurophilum, and then moved into studying pathogens like tuberculosis and leishmania. But the interactions between the pathogen and human or the pathogen and plant, um, that seems like an incredibly like studying the pathogen alone is hard. Studying the human alone is hard. Studying the two together seems like just incredibly challenging. And so was that part of what was exciting to you was just the extreme unknown?
I think so. I think for sure, because you can understand one side and understand, like we know infection occurs, we know disease occurs. And during my PhD, I studied the pathogen and I really focused on that and how it adapts to its environment. And then during my postdoc, I was in a systems immunology subgroup. And so that really started to bring about the host side of things. And whether it be in a crop or in a host, like a human host, the concepts are very similar. You have your innate defenses, you have your adaptive defenses, all sorts of mechanisms at play. And I think gaining that new knowledge of immunology and kind of being empowered to understand the host perspective, that's what really like led me to get to where I am now, looking at both the host and pathogen. And you're absolutely right. One of them is challenging, so two is going to be even more challenging. So I think you can learn a lot of things.
Yeah. Well, so maybe let's actually just go way back in time instead of talk about Cryptococcus neoformans and or however one is meant to pronounce that properly.
Yeah, you got
Oh,
it.
great, fantastic. And so tell me a little bit about how you came to study that particular pathogenic yeast, why it's an important thing to study. And walk me back there.
Yeah, absolutely. I think from my undergrad and my master's, that's where I studied fusarium, the agriculture side. And then for my PhD, I wanted to shift. I still wanted to study fungi, but I wanted to look at the human relevant pathogens. And so at the University of British Columbia, Jim Cronstad was a researcher there studying Cryptococcus. There are not many fungal researchers, and so I reached out to him, and I was very passionate about studying, and so he invited me to join his lab. And I think it's an amazing pathogen because it's quite fascinating to study, and it has global importance and relevance with infections around the world and with immunocompromised individuals, and kind of that the importance and dynamic that the host health plays on whether the fungi is a pathogen or not was a very intriguing question for me. And so that's what led me to study that pathogen. And then when I started my own lab, I was able to bring the two of them together in the sense that, I'd say half or a third of my lab studies crop relevant fungal pathogens, and then another two-thirds study the human relevant fungal pathogens. So I didn't set out to, kind of have those two arms going within my research program, but it's worked out beautifully, and it allows me to kind of keep all the things I found interesting along the way, I can keep going with them.
That's great. Well, so maybe for those of us that are sort of new to this space, can you give me the headline kindergarten explanation of why we really need to understand host pathogen interactions and why studying host alone and pathogen alone isn't sufficient?
I would say that it's really important for so each individual has their own immune system, their own way to respond to infections and the environment that surrounds them. And that's based on genetics, it's based on, what you're exposed to on a daily basis, many different factors. And so a pathogen does not know that. A pathogen does not know what the host is going to be like, right? It's not a relationship until they become together within the host environment. And so I think that's why is because each system is its own operating component separately. And when they come together, you can either fight it off and be healthy and get over a cold or get over an infection because your body is built to do that, or you don't. And for those that do not, then there's a lot of implications downstream and healthcare costs and quality of life compromising and compromisation. And so I think understanding that a pathogen is one aspect of the picture, the host is the other aspect. And when they come together, we don't know the answer. And each individual has a different mechanism or a different way to respond. And so that means that we can learn a lot from every individual's situation and response. And I believe that's part of why it's important to study both because we can make treatments on a population basis, but if it doesn't work for one individual or one pathogen, it's not really that useful. And so we need to be able to understand in many different layers.
That makes a lot of sense. One thing that I'd like to understand a little bit more is when you think about pathogens, and I realize much of your work is on fungal pathogens, but uh, there, what really is differentiating a pathogen from a helpful microbiota that is just part of our life?
Yeah, yeah. So that it's a great question, and it really ties into that host pathogen interaction, right? Because for Fusarium and Cryptococcus, they're not typically within the host, and so you can become colonized with them upon exposure from the environment. But if we consider bacteria within our gut, or fungi within our gut, or microbes on our skin, of course, we're not constantly infected with them. So what is that trigger? And there is a lot of research that goes into understanding how do they survive together and how do they adapt? And when is that dysregulation? So, one of the examples for fungal pathogens that I often think about is that if you're put on antibiotics for a bacterial infection, of course, then there
Mhm.
might be side effects that come along. Oral thrush is a common one where you have yeast that are able to grow in the absence of bacteria. And it is a real tangible observation of the competitiveness that goes on between microbes in our body. Those microbes exist all the time, and yet we do not have the symptoms the same way all the time. So it's this balance and it's disruption to that balance, and that's where you get the response. And hopefully, within a healthy individual or an immunocompetent individual, your body can bring you back to that equilibrium. And again, each person has their unique equilibrium. So it's fascinating to study the interactions of microbes together, and then that switch to a pathogenic phase, and as well as how external factors like antibiotics can impact that.
Yeah, that's really interesting. And how much, when we think about interactions on a macro scale, we're often talking about ecology, talking about, large animals interacting with other large animals. How much, from your perspective, do those principles of ecology or systems ecology come out when studying these questions of biota interactions, biota host interactions?
I think I want to go with the principle. So my research program is built around a One Health concept. And when you talk about ecology and you talk about these macro scales, that's what comes to mind.
Mhm.
Because the principle of One Health is that you're looking at the interaction across human health, animal health, and the environment. And so you might be studying one microbe that is important for a crop to thrive or within the soil, something like that. But how is that microbe impacted by the whole environment? And so that's what I think about when I think about ecology. And one of the examples that ties into the work we do is looking at fungicides that are applied in the field to prevent fungal infections of wheat, for example. Those fungicides then reside in the environment. And if we look at that on a grand scale, the microbes within the environment are exposed to those chemicals. And if it is a fungal microbe that is then, infects a human, for example, we may be unable to treat it, or it might be difficult to treat because it's already been exposed to these fungal drug or antifungal agents or fungicides. And so kind of that whole systems approach as to how, something that we apply to the field so we have better food and food security and safety can actually harm how we treat infections in the clinic. And that big picture concept is it's nice umbrella that helps us to stay with the vision of what we're doing and how important that minor discoveries in the lab can really have implications far beyond that.
You had this really lovely paper in just maybe 2023 on reviewing this concept of One Health. And so maybe walk us through a little bit where that concept emerged from. When did you first see it? How did you first interact with it? Um,
Mhm. Yeah, I would like to say that it was intentional, but it was not. So, what happened was is that yeah, through my training when I was studying agriculture health and fungal pathogens in agriculture, and I bring that into my lab, and then I'm studying human fungal pathogens in the clinical side, and I brought that into the lab. It was very much so that I could pursue multiple avenues for funding. You can have industry partnerships. It was very strategic in that sense, but they were two very separate programs. And then being at Guelph, One Health is a term that they really promote and advertise, but it's a very difficult to grasp term because a lot of people either feel excluded because they're not doing One Health research, or it can be defined in like a million different ways. But that's also kind of the beauty of it. And so with the flagship and Guelph, Calgary is also doing a lot of One Health. There's institutes in Switzerland. It's becoming more of a globally known term. And so when I was reading about it and this review kind of came up and we were looking at antifungal resistance and realized that these fungicides in the field, these azoles, are what we use commonly to treat cryptococcal infection. And it was kind of this serendipitous aha moment that I was like, oh, my whole program can like center around this one health umbrella. And I remember pitching that to some of my colleagues, and they were like, "Oh, that's a good idea." And I'm like, "Yes, it is, isn't it?" But it was very much from the literature and reading what's been done and trying to find our own niche that it came about.
That I love hearing these stories of serendipity because the there is so much you're receiving inputs constantly and influencing your perspectives. And so this sounds like a place where you really were able to synthesize multiple inputs to come up with a really unique thesis.
Thank you.
I'd love to dive in. At the core of a lot of your research is is proteomics as part of these studies, even going back to that sort of first some of your first work. And I'm curious, why? You could you could study any ohm you like. You do in fact study many ohms together. But why did you choose to focus on proteomics in this space of host pathogen interactions?
Yeah. I probably do a lot of self-reflection on this question for some I don't know why, but for some reason, I often think like, why proteomics? And I absolutely love it. And I it's the technology, it's the discovery that goes with it, it's the computational advances. Like it's just something that has really grasped me from the very beginning with research. And I remember I started doing 2D gels. That was the first introduction to proteomics. And they were so difficult. And I remember like endless times washing the 2D gel plates, and you'd come in the next morning when they're dry and they'd have like soap residue on them, and you'd have to start all again. And it would be a day, and the electrophoresis wouldn't run properly, all these things. And when I think back to those days, I'm like, why did I ever keep going with this? Like, it doesn't make any sense. But I think it was the fact that I could tangibly see something on a screen, like the dots from the 2D SDS page gels, and then measure those on the mass spec and get an idea of what is going on biologically. It was that aha moment for me as a student, because everything else had been textbook or in a lab where it's very set up for you and the question is there. This
Mhm.
was the first time that I was able to kind of discover on my own. And so I knew that no matter which questions I was asking or which pathogen or host system, proteomics was the technology that I wanted to use and apply. And that's what I've done.
Wow. No, that's great. It's amazing how these early choices can shape our path, because
Yeah.
it's possible that your very first technology might have been a fish assay or
Yeah.
something like that. And then you would be doing transcriptomics for the rest of your life. And,
Well,
and so,
that's oh no.
uh, uh,
Yes.
well, I guess just going down this theme, I'm curious, is there a question that you've encountered throughout your career where you look at it and you're like, you know what? If I had tried to answer this with doing genetic knockouts or looking at transcript, I wouldn't have been able to. The proteomics in particular was critical for answering this question.
Mm. Well, I could say that about any project, especially when I'm writing a grant. That that would be easy. But I would say is the so we use proteomics a lot for discovery in my lab, and then we follow it up with a lot of biological findings. So genetic knockouts, as you say. Sometimes we have to do RNA and qRT-PCR transcriptomics. It's not my favorite, but we do it sometimes to validate and see different aspects. And so for me, if we don't get kind of a thread of interest of where to start with proteomics, then we change the research question. And that's because I think there's a lot of power there in looking at the proteins. But that being said, with some of our biggest studies, one recently we were looking at antifungal resistance. We also are very cautious to not say proteins are the most important or the only aspect. We also do whole genome sequencing on our different isolates. We may look at transcript changes across to see if it's a consistent change in the protein or a post-translational modification. So we're like always open to using complementary approaches to further our argument, and whether it be that the protein was the best way to do it or not. One of the most recent applications where proteomics really empowered our research, I would say, is a paper on the blood proteome and looking at diagnostic markers of cryptococcal infection. And we wouldn't be able to find that with genomics or transcriptomics. And the idea of where we want to take this is into, real world diagnostic approaches that are affordable and non-invasive, because right now a lot of them require spinal fluid taps and collection of cerebral spinal fluid to diagnose infection. So if we can use proteomics to identify potential proteins for an ELISA assay that we could apply, that would be
Mhm.
really empowering for our group, but also for how we detect the pathogen and the infection.
Yeah, that's a really powerful example. One of your earlier studies was looking at the secretome, and that's often a place where transcript simply doesn't know what is secreted or how that's changed. And so I'd love to hear, what is your favorite study that you've done where you're like, wow, I I I I understand that all the omes are important. I'm a systems biologist also. However, every now and again you look at something and you're like, wow, that the protein was the key driver of X. And
Mhm.
so I'd just love to hear what your favorite example of that is.
That's a good question. It's like picking your favorite child.
I know it's very challenging.
You do always have a favorite, so it's okay. But no, I would say we had our recent story on antifungal resistance is probably the kind of the most exciting in a sense, but also like the most intriguing because antimicrobial resistance, antifungal resistance is often tied to genetic factors, the mutations in genes, and that is definitely a driving factor of it and how the pathogen evolves upon exposure to drugs. But it's not always the case, and there's a lot of heterogeneity in resistance. So we wanted to use proteomics to look at antifungal resistance in Cryptococcus, and we identified, I think, 6 proteins that were higher in clinical isolate and our lab evolved isolate. And so that was a really cool moment where I thought, like, this is neat. We never would have seen this. And it's also not consistent. So, like a gene mutation stays once it's there. It usually stays unless it evolves differently, but the protein level would go up and down depending on exposure to the drug. And so it shows you the dynamics of a system and the dynamics of that resistance. And so we showed that if we inhibited the protein, we could actually reverse resistance, which was really cool. And we
Wow.
could turn it on and off. So we could have the strain that's resistant, inhibit the protein, then it's susceptible. And then we take, change the media it's growing in, and now it's resistant. So
Mm.
just that new understanding and kind of that new idea of where proteins can really play a role. That's my favorite one so far.
That's really amazing. And the dynamism, I think that's one of the aspects that's so special about the proteome is that it it operates at multiple different timescales. Things like truncations or degradations, PTMs are very fast.
Mhm.
And then you have turnover mediated things that require translating a protein, and that can take a very long time for the signal to make its all the way back again. And so those cycles that you're describing or changes per environmental context dependent behaviors are so neat when you see them in the proteome.
Mhm. Mhm.
I guess maybe carrying this on further, just thinking about you've done a lot of work in metabolomics and gene expression at others. How do you think about integrating the different omes together? This is every lab has their own style of a way that they try to think about how the omes interplay, how they try to bridge the data. How do you integrate the diversity of omics technologies together?
Yeah, it's challenging, right? And I think every lab that does systems biology or does that integration, as you said, has their own way of doing it. And that's usually through a lot of trial and error. So you had mentioned the molecular farming study, and that was one of the first ones where we integrated the two approaches. We had proteomics of the plant, and then looking at metabolites as well that were produced.
Mhm.
And what was funny, not so funny for my PhD student, but kind of funny in hindsight, is that, we have the proteome and we can identify thousands of proteins. And he was like, oh, what happens when I get so many metabolites and I don't know what to follow up on? And I think in the end he got 14 that he was like looking at. And so we were both very deflated
Yeah.
because we thought we're gonna have this like keg map of all these pathways and we're gonna do all this amazing stuff. And then he's like, I can just do this manually. And so we can like, annotate it. So I think the reality of systems biology is immense and enormous and exciting, or maybe the thought of it is, and then the reality of it is that it really comes down to your question and what you want to find. So
Yeah.
for some things, looking at one metabolite can be enough because it's a promising antifungal candidate that you want to pursue.
Mhm.
For others, you want to understand the whole pathway and how those compounds interact with others, and
Mhm.
that's much more dynamic. So I think bringing the two together is a very conscious thing that we do from the outset of a project. It's like this is going to be a metabolite focused project and we're going to complement it with proteomics. Or it's going to be proteomics all the way, and we're going to see if we get a compound that we're going to then test out and see what the target is. That helps us to bring them together in kind of a sensible way,
Hmm.
because otherwise it is very challenging to mold it all together into a story that makes sense. And
Yeah, I'd love to double click on this aspect of molecular farming. Just how much can we do with molecular farming, biofarming?
yes, I would I'm not an expert in the area. I would say this was a really neat opportunity to work with an industry partner. When I first started my position, they wanted to do proteomics, and being new, I was like, yes, I can do that, no problem. And so we set up a project, and it was a 4 or 5 year project, this collaboration. I had a PhD student working on it. And it was not something I had ever explored before. I had no idea what they were doing. And the concept is it's, again, taking this whole systems level approach in the ecosystem, is that within the natural environment, agrobacterium bacteria infect plants, specific plants, and they cause these tumors. And that's by they inject their own DNA into the host, and then they have the host produce proteins that they need to survive. And so it's this, fundamental concept that occurs in nature. And so there's scientists that are then harnessing this, as we do for many different kind of scientific innovations. And so, but you could have a plant produce an antibody, for example, or a biologic, or, another compound that you'd like. And within a plant system, you also are not, you don't have the same considerations perhaps as with, like I'm thinking lifestyle choices, like veganism, for example. They may not want to use components that come from a cell system or an animal model. And so using the plant model, it's a different opportunity there. And so companies that are doing molecular farming, they want to produce an antibody, inject it into plants using a natural process of this bacteria infecting the plant, and then you collect the leaves, extract it out. It's a very crude product, and then you can purify it. And so we were using proteomics to kind of understand each level, each layer. So, how's the best way to grow the bacteria? What happens when you infect the plant? How do the bacteria respond? And then how much product you get at the end, but how could you improve that output? And that was our question with the paper, was really, can we help companies improve their molecular farming pipeline and get better yields?
Mhm.
And so we identified proteases from the plant that actually degrade the target antibody. So we showed just a proof of principle. It was a very early study for us to show if we knock down these proteases, or one in particular, then there's more antibody produced.
Mhm.
And a particular example was a breast cancer drug, trastuzumab is the one that the company was using as their flagship.
Mhm.
So it was such a neat way to, kind of look at this natural infection, but then how can we use it for health
Mhm.
that I wasn't aware of before this
It
opportunity.
seems miraculous. One of the things that really stood out for me in the paper was that flask grown versus bioreactor grown bacteria actually had an impact on the plant uptake behavior.
Yeah. It was really cool, isn't it? It's so neat because the bacteria are primed to infect if they're growing in the bioreactor. So it's this neat idea that they're ready. So when you put them in the plant, they just like go for it and they can do the infection really quick. And yeah, it's not something I'd ever thought you'd see a difference of, and how proteomics would allow us to see that difference. It was very discovery and neat to see.
Yeah, it wouldn't, I mean, naively, it makes sense post facto that these environmental factors change the proteome. We see that in cancer all the time, but in a bioreactor environment is different than how pathogens might grow naturally, microbes might grow naturally. So I wonder also if there are other forces beyond what's secreted. Maybe there are even things like density-based effects, mechanical effects that drive those changes in behavior.
Mhm, mhm. Absolutely. And like we only studied a few of them, right? Like we had a very specific bioreactor system and then a specific flask system, and just compared a few parameters, but you could absolutely expand it, and the size of the bioreactor would also likely, right? Because then you have different quorum sensing and all different components. So yeah, there's a lot to be done.
That's really neat. Well, so I'd like to change gears a little bit. We've talked about different omes and proteomics, and I'd like to put you on the spot a little. And the technologies we have today are amazing and they're powerful, but what is something that you wish you could do with your available platforms that you can't do today,
Hmm. Hmm.
or that is
Hmm.
hard today?
Yeah, yes. I think one of the biggest things would be accessibility of instrumentation. So if I think about for my own lab and you think about buying a mass spectrometer and state-of-the-art mass spectrometer and you're spending a lot of money on these instruments, the odds of having an undergraduate student being able to use that instrument or even a grad student being able to use it are very low.
Mhm.
And I know for myself, I did not have hands-on on a mass spectrometer until I was in my postdoc. And so I learned so much within the first week and I gained so much confidence and more knowledge of how things were working and what a peptide actually meant uh, in that week than I had in years. And it was because I could actually touch the instrument, change the capillary,
Mhm.
understand when things were really hot, like simple stuff. But that's really hard when we have instruments that are so expensive, and that puts a lot of pressure on them operating all the time. And
Mhm.
so I think it would be nice to have, you buy a big new shiny instrument and then you get like an older model that would be for testing and for training, and it's like a two for one. That would be what I would ask for, is so that you can actually have everyone in your lab hands-on training on that piece of equipment. So when they want to go get a job at a company, they can say, I've worked on your mass spec before. That would be really empowering.
Yeah, no, that's really interesting. That's an aspect of the conversation that doesn't come up very often, which is just the accessibility of the tools and availability of the tools. And I'm curious actually just to go a little bit deeper there because you mentioned as you got hands-on with the mass spectrometer, it changed your understanding of the workflow and process of proteomics. Can you just say a little bit more about that? Because I think this is a place where oftentimes people coming from other domains, they their assumption is, okay, well, you walk up, you press go, and proteome comes out. And that's not actually what happens. And there's a lot of detail and understanding. And so I'd love to just go to that moment in your life where you weren't reliant on other folks as much and you were there face to face with the instrumentation yourself.
Mhm, mhm. And you're absolutely right. It has a huge role in, how I see things and how I do things, how I teach now, whether it be, at the instrument or it's theoretical. Because when I didn't have access to the instrument, it was very much a black box, and I never thought that I would be able to understand it. I never thought that I'd be able to explain it or know what's going on. And it's not because people wouldn't teach me, but it was because I had no visual connection to it. So I remember early studies, you mentioned about the secretome,
Mhm.
and there were some aspects of targeted proteomics in that assay. And I remember we were supposed to look at quadrupoles, but it meant nothing to me, right? And these peaks, they meant nothing. And so it wasn't until my first week during my postdoc when I was actually allowed to clean an instrument that I saw a quadrupole, and I was like, oh my goodness, it's exactly what it looks like in the images, and this is how it works. And it really helped it click. And I think that it works on both sides because yes, it's a very, high-end instrument, and proteomics does require expertise to some level, but really anyone can gain that expertise, and especially now instruments are very automated. So it can be a push this button, but you still want to know how it's working and what it's doing. And I think the other aspect of that is the mass spectra, the de novo sequencing, and understanding what a mass spectra is. That's always been a very like amazing aha moment for me when it came to mass spectrometry. Is it's like these peaks and what do they mean, and you can actually interpret it. So I think empowerment, training, all of those aspects really make proteomics more appealing to more people if they know they can do it.
Yeah, that makes great sense. And maybe just one more digging in a little bit deeper here as well, just about the data that you're able to produce, the questions you're able to ask. Where are the boundaries of there? If you could invent your magical perfect system to answer questions that you're like, today it's just difficult. Um, what would that be able to do that you can't do today?
Hmm. What is difficult now is probably the entire pipeline. Like, so if I'm thinking about drug discovery, for example, and if I think about all the different layers that we use in my lab and all the different aspects, and so if you use like computational ways to predict or design drugs, and then you want to synthesize them and produce them in the lab, and kind of each of those is its own individual piece, but it would be really neat to have it all together in one workflow and have it so that it's like a given. It's like an automated system, right? Where you have this amazing mass spectrometer, you have your data analysis tools, you've got the upstream, the downstream, all together in a pipeline. And that is my kind of dream of what I would like to build and incorporate. That would all of the little barriers that exist in each of those pieces would no
Yes.
longer be there. It would be one
Yeah,
piece.
just a whole integrated.
Yeah.
I think that's definitely something that we've seen in our lab as well, is that there is a lot of domain knowledge that's required in switching from X tandem to uh, pick your favorite sequence analysis tool. Uh, they each behave just a little bit differently, and so understanding how they connect together, there are a lot of little places where there are just tiny little friction points along the way that sort of coalesce into adding up into barriers to having more and more people joining us in the field and saying, yes, proteomics is a go-to tool for me. Switching gears entirely, let's talk about HUPO, and then I want to talk about moms and proteomics. What are you most excited about HUPO that your lab is going to be sharing with the rest of the world at this conference?
So I'm very excited for HUPO to get started.
Yeah.
It's a lot of meetings and preparation for it and a lot of anticipation of the conference itself. One of the most exciting things is that I have multiple people from my group coming from the agriculture health aspect, which they would typically not ever go to HUPO, and I would not think to send them to HUPO. We have different conference proteomics, plant proteomic conferences that they would go to and more biologically relevant agriculture ones. And so I'm super excited of the breadth that HUPO is bringing this year with, and we have the theme One Health powered by proteomics, but really having that take effect and come to play where you have sessions dedicated to environmental health and session to clinical health, the integration with veterinary and animal health as well, and making sure that everyone still feels like it is HUPO and that it's, the HUPO they know and love, but we're also bringing in new aspects and new people. And I think that's super exciting.
That's really neat. I mean, expanding the tent to your
Hmm.
point earlier, all these things that live in agriculture affect humans, and vice versa. So that concept of bringing these pieces together is definitely a really exciting one, and an opportunity also because it's closer to your backyard this year.
Yeah, for sure.
Um, and I guess, are there other stories from other labs that you've heard snippets of that you're hoping to see more of at HUPO this year?
Hmm. I think definitely the computational side. Last year at HUPO, those rooms were not even standing room only. They were out the door only.
Yeah.
So I think the hearing the latest of what people are doing with computational platforms and AI, of course,
Mhm.
there's different chat bots now and agents that are supporting proteomics analysis and visualization. So that side of it, I think, is really cool. And I'm definitely looking forward to seeing that and also having my students see the components that are changing. And the other aspect I would say is being able to kind of showcase the breadth of what's the technology, of where things are at and where they've been, where companies are going, pushing the limits. So I enjoy going to the sessions as well by sponsors and industry partners so that you really get to see like what is happening at the next stage and what can we look forward to.
Yeah, that's great. Well, and just HUPO is such a distinctive meeting. Uh, for you, what is it that you like about HUPO? What's your favorite part about it as a conference? There's so many conferences in the world. Why is HUPO
Yeah.
great?
Yeah, and each conference has its own place for sure, and its own, I think with HUPO, it's the fact that it is a large conference. It's our largest proteomics conference that we have globally, and yet it feels like a small meeting. It's a very connected network, very collaborative. You get to see friends, you get to see colleagues from all over the world. I love that it's so international, that it rotates across the regions, that you have so much. It's so consciously diverse. And that's not something that we typically see. And it exposes you to all different science and researchers and also gets feedback on your own science and exposes what you're doing to the world. Just a great platform.
My last question in this line for you is about moms and proteomics. For folks who aren't familiar, what is moms and proteomics? Where did it come from? And then I'd love to hear more about what Moms and Proteomics has planned at HUPO.
Yes, for sure. Moms and Proteomics is an international initiative and now an international community. We have close to 250 members from around the world, and it's very much dedicated to supporting and recognizing mothers and women in STEM. And it came about actually from HUPO 2021. There was a call for hot topics in proteomics. And at the time, we were coming out of the pandemic, and I had two young children at home. And I was like, you know what's a hot topic? Caring for children while being an academic. That's a hot topic. And being a mom and trying to navigate everything. And so I pitched this idea to the workshops, and they were like, I don't know. We were thinking like technology and things, and I was like, trust me, we can do this. And so they really gave me that first opportunity to build it into something and showcase it. So it was very much a grassroots, bottom up community building. And since then, it's I've had different events, different activities. We actually had a virtual coffee networking event this morning, and now at HUPO, it's part of the program. And every year we have different sessions. And so this year, I'm very excited. We have our first time. We're bringing an invited speaker to discuss resilience building and inclusion within the mass spec, the proteomics biology community. So it's on the program, I think, for Tuesday, included with registration, and I think I'm really looking forward to it.
Oh, that's so exciting and very inspirational that you had this notion and were able to bring it into the world, and now it's a thriving community. I think that speaks volumes also just about how welcoming the proteomics community is to new ideas, and that you can say, you know what, hey, this is hot. Let's do this. And it's a huge important part of the conference. So for people who haven't been there, it really has become a very central part of HUPO, and something that I'm so delighted is supported. My last question for you is really very forward looking. I'd like you to put on your future you hat, and you're 10 years from now, 20 years from now, what is the thing that you look at then? And you're like, okay, this is what I've learned. This is what I've been able to accomplish. This is how proteomics has shaped my world.
Yeah, I think looking into the future, I would like to, in all of our grants, when we write, we're going to change the world, we're going to make a difference. I would like that to actually be true and not just be a statement that I include the last line, and really translate some of our early findings into tangible applications. And whether that be in the agriculture field or whether it be in the clinic, like entrepreneurial startup company, and supporting my students through that journey as well, because many of them are very interested and passionate in industry and translation, knowledge translation. And so I think looking forward that, yeah, moving things from discovery into where we actually produce it or we provide this translatable application, that would be the probably the most rewarding component.
Well, I look forward to seeing that come into reality. I totally believe that is coming, and we're seeing it all over the place throughout our communities. So thank you again so much. This has been so much fun. I'm really grateful that you were able to join us today.
That's wonderful. Thank you. I really appreciate it.
We hope you enjoyed the Translating Proteomics podcast, brought to you by Nautilus Biotechnology. To contact us or for further information, please email translatingproteomics@nautilus.bio.